Hi everybody, it is your AP Biology teacher, Mr. Poser. Today we're doing a Unit 4, the cell cycle and cell communication, or cell communication and the cell cycle AP Biology recap. We're going through all 7 topics of this unit. I'm going to try and get through them sufficiently fast. Okay, I'm going to try to go over the basics of each one of these 7 topics. Ready? 4.1 is on cell communication. 4.2 is on introduction to signal transduction. 4.3 is signal transduction. 4.4 is changes in signal transduction pathways. There's a theme here. 4.5 is feedback, 4.6 is cell cycle, and 4.7 is the regulation of the cell cycle. All right, so the first 5 topics are really all about how cells communicate with each other and why cells communicate with each other. And then 4.6 and 4.7 is about the life and the reproductive, more importantly, the reproductive cycle of a cell. So let's get started. 4.1, so this is about cell communication, right? And if you are a cell, okay, if you're a cell that's part of a multicellular organism, like say a human being, or if you're, an amoeba or like a euglena or a little bacterial cell, okay, you still need to be able to communicate with other cells in your environment, or you need to be able to communicate with other cells as part of the tissue that you form up so that you can form this functional organism, right? So cells have to generate, transmit, receive, and respond to chemical signals. It's part of the requirements of being a living thing, being able to respond to things in your environment. And cell communication is one of those factors. Okay, so all cells need to be able to communicate, and like I said, they must be able to communicate in order to function, and that could be done through local or long distance signaling. Local meaning in the area, long distance meaning, well, not in the area, long distant for a cell, right? So the first local signaling is called direct contact, connecting plasma membranes and cytoplasm between adjacent cells. When two animal cells connect their plasma membranes and their cytoplasm in order to communicate through direct contact, it's called a gap junction. And plant cells form what are called plasmodesmata, and they're slightly different because a plant cell has a cell wall, an animal cell does not. Okay, so a plasmodesmata is kind of like, or plasmodesmata are kind of like channels through which cytoplasm travels through to adjacent plant cells. Okay, another factor in direct contact signaling, you may or may not have heard of these. They're called antigens. And antigens are membrane proteins that allow cell communication, and they are important for immune function. So your immune system, all right, I don't know a ton about the immune system, so I'm trying not to go out of my depth here, but basically how your immune system functions is it will recognize certain pathogens or certain types of cells, foreign substances that should not be in your body based on, or I shouldn't say substance, foreign cells that shouldn't be in your body based on the antigens that those cells have. Okay? And they're kind of like signaling marker proteins on the surface of a cell. And say, for example, it's a bacterial cell that doesn't belong. It could be, pathogenic. Your immune system will recognize that antigen. Okay, I'm making this sign here because it's kind of like a little signals on the cell membrane. It will recognize that antigen is not belonging in that organism, and then it'll take care of it. It'll kill it or whatever, swallow it up and digest it. So that's what antigens are, and that's another form of direct contact communication. Local signaling. All right, we can categorize direct contact as local signaling, but we can also categorize paracrine, autocrine, and synaptic signaling as local. Okay, and then hormonal signaling or endocrine signaling is what we call long distance, and there's some pictures here that I'm going to walk through right now. So autocrine signaling, which is a type of local signaling, a cell receives its own signal, and some positive feedback loops are done through autocrine signaling. So a cell will send its own signal, and it will also receive its own signal, and that will cause a cellular response. We're going to talk about that more in a sec. Paracrine signaling is when one cell secretes signals to nearby targets. An example of this is histamine, which is an allergic response. Okay, so when your body or when a tissue becomes like inflamed, that is usually because of histamine, and that's an immune response to whatever's going on. So histamine actually causes sneezing, right? So something enters your nose, it triggers a cellular response. It binds like a substance in your nose, binds to a receptor on what is that called? An olfactory receptor cell. And then, if it's something that the cell doesn't like, okay, it'll send a signal to its neighbors through histamine, and it'll cause you to sneeze eventually and get rid of whatever it is that's bothering your nose. And then long-distance signaling is what's called endocrine, and you have a system named after that. You have an endocrine system full of glands that release hormones through your blood and through your lymph to communicate with distant parts of the cell, right? So, for example, your pituitary gland is going to release hormones that are going to tell, they're going to tell you to go through puberty, pretty much, to put that simply. But the point is, okay, one cell tells a very distant cell what to do through endocrine signaling. Okay, and then the last kind, this is another type of local signaling. It's called synaptic signaling. Super important because it's how your brain works. Neurons are releasing neurotransmitters into a synapse, and a synapse is a gap between a neuron and another cell to stimulate target cells, right? So if a neuron is picking up a signal, or it'll have to send a message to another neuron, or if you're a motor neuron, you're going to have to send a signal to an effector, and we're going to talk about effectors here in 4.5. And it'll have to tell that neuron will have to tell that other cell, that effector cell, what to do, and this is called synaptic signaling. All right. So that's how your nervous system operates. Let's talk about 4.2. So we are talking about signaling, right? Cell signaling can be boiled down into three main steps. One, a signal has to be received. That's a reception. Two, the signal has to be brought into the cell so that three, the cell can respond to that signal. So 3 steps of cell signaling are reception, transduction, and response. And we're going to briefly kind of go over those in 4.2 and in 4.3. So reception begins when a ligand, that's right, ligand, binds to a receptor. Okay, so in the last unit we talked about how enzymes act on substrates. A receptor binds to a ligand, and a ligand is a signaling molecule. So let's say, for example, you take some ibuprofen, and that will bond to certain, and that will be digested and put into your bloodstream. And then that ibuprofen floating around in your bloodstream is going to bind to certain receptors on cells and cause a cellular response, right? Or any kind of medicine for that matter. So a ligand is very particular to what receptor it binds to. And a receptor, as I put here, is a protein in the cytosol or on the plasma membrane of a target cell that binds to the ligand. It receives the message. The ligand is the message, and the receptor is, receives it. Okay? And ligands and receptors are very specific. They kind of fit together like a lock and a key, right? A key only opens one kind of lock in particular, and a ligand only binds to one receptor, kind of similar to an enzyme and a substrate. There's different kinds of receptors, all right, and I'm going to briefly go over these. An intracellular receptor is inside of the cell, and the way that a cell usually responds to an intracellular signal is through gene expression, which is all about Unit 6. Cell surface receptors, which are the most common. Ligand-gated ion channels. Ooh, this is a fun one. Ion channels are, membrane proteins that allow ions in and out. Think back to Unit 2. And if it's ligand-gated, that means the ligand is kind of like a key. A ligand binds to that ion channel and opens it up. It's like a key opening a door. It's amazing. And most importantly, well, maybe not most importantly, but maybe for AP Bio, it's the most important, a G protein coupled receptor, aka a GPCR, works with an intracellular G protein, and that's a special kind of receptor that we'll talk about more in 4.3 when we talk about the epinephrine signaling pathway. So once the signal is received, a message can be transduced. This is step number 2, or carried from one molecule to the next to produce a response. All right, so this is a more complicated image than what you're probably going to have to deal with on the AP exam, right? But it's showing here that, okay, here's a ligand, it binds to a receptor, and then a series of steps happens to bring that signal into the cell to cause a response. This right here, okay, where it brings in that signal from the outside to the inside, that is called transduction. And there are many different what we call transduction pathways that can occur. You could spend your whole life studying signal transduction pathways and cellular responses to different signals. Okay? Different steps in a transduction pathway provide opportunities to control or alter a cell's response to a signal. So like I said, these can get very complicated with a lot of steps. And when something has a lot of steps, there's a whole lot of ways to kind of like screw with the whole process, right? So you can mess something up big time. In multiple different ways if there's a long process that involves a lot of steps. You mess up one step and it can, it can bunk up the whole process, all right? So we're going to talk about that more in 4.4. So how transduction occurs a lot of times is through what's called phosphorylation. Hey, we're familiar with that. Adding a phosphate, right? Making ATP from ADP, right? That's phosphorylation. And this should be somewhat familiar. Phosphorylation is also what's used to kind of activate a protein. Hey, turn on this protein. It's like an on or off switch. When it has a phosphate, it's on. When it doesn't have a phosphate, it's off. Okay, and when something is transduced, when a signal is transduced, a lot of times it's a phosphate getting passed from one protein to the next. So say, hey, go do this. Hey, go do this, to make that cell function and respond to a signal. The proteins that often carry those phosphates from one protein to the next are called kinases or protein kinases. And kinases are going to be activating other proteins. Protein phosphatases do the opposite. They turn off. Okay, so kinase activates another protein, a phosphatase deactivates it. It removes phosphates and it stops their function. And what these are called, okay, when you have multiple kinases and multiple phosphatases kind of functioning together to send a signal from one protein to the next and cause a cellular response, that's often what we call a phosphorylation cascade. And a cascade is like kind of a flow down. Yeah. Okay. Another important molecule in signal transduction are called second messengers, and they can amplify a signal in a cell, causing multiple pathways. We're going to look at an example here in a second, but they can, or in other words, I like this word, they can broadcast a signal throughout a cell, right? So if a cell needs a lot of different responses to one signal, okay, a second messenger could be used to spread that signal throughout the cell. And the most famous one, one that we're going to be talking about is going to be CAMP, cyclic AMP, and which is derived from ATP. And here it is, adenylate cyclase or adenylyl cyclase is going to break down ATP into CAMP. And this is a signaling molecule. This is ATP. Calcium is another good second messenger too. Okay, but step 3 here, again, transduction of a signal results in a response. And here's possible responses. The cell could grow, it could divide, it could secrete something, right? Produce a protein and send it out. Gene expression, okay? It could activate make another protein or a secretion, could be the release of a lipid, whatever, okay? And then apoptosis, which is like, all right, programmed cell death, like your contract at this company is terminated. Those are all the different responses that a cell could produce from a signal. Here's 4.3. As I was saying before, there's a lot of variability in signal transduction between pathways between a variety of receptors, ligands, second messengers, and phosphorylation cascades. Again, you could spend your whole life studying signal transduction. It's crazy, right? The variety of responses and signals and transduction pathways is ridiculous, right? So we're going to study like one, okay, for AP biology. Epinephrine, aka adrenaline, signals a fight or flight response. So if you've ever heard of, epinephrine and adrenaline, what they do or what it does, okay, you are in a situation where you have to fight for your life or run for your life. Your adrenal glands above your kidneys are going to release a whole bunch of epinephrine, and that's going to tell cells what to do here. It's going to increase your heart rate. It's going to stop digestion. It's going to increase your breathing rate. It's going to increase your awareness, right? And it's going to get energy ready to go for you. We're going to talk about that in a sec. Okay, but here's some other examples of signaling. Quorum sensing is when bacteria release signals that alter gene expression and fission of other bacteria when their concentration increases. This is an example that was brought up in the AP Biology course and exam description, so we're going to talk about it here. But yeah, basically bacteria divide, all right, and the more they divide, they release a signal, and if there's too much of that signal and they collect too much of that signal in their cells, they stop dividing. So it controls how concentrated bacterial colonies get. Yeast cells receive pheromones that trigger division. Yeast cells will send each other signals that will cause them to divide. Here's some other examples of signal transduction and responses. The expression of genes on the Y chromosome triggers pathways that initiate male development. Okay, so your Y chromosome contains a whole bunch of genes that, well, will make you a biological male, right? So when those genes are expressed, it'll send out signals that'll start to tell cells, particularly in the reproductive system, to like, hey, start producing stuff that guys have. So that's another example. And then one more that we're going to talk about is ethylene. Okay, ethylene is produced by ripening fruit, and ethylene will cause a signal transduction pathway that will result in more fruit ripening. That's called a positive feedback loop. We're going to talk about that in 4.5. I know I keep saying that, but like we will, I promise. So what we're going to briefly talk about here to see these kinases and second messengers and receptors, GPCRs in action, we're going to talk specifically about the epinephrine pathway. I shouldn't say specifically because I go over it specifically in the 4.3 video. So if you want specifics for it, go there because I'm going to kind of gloss over it in this video. But as I said before, epinephrine triggers a fight or flight response. It gets you ready to fight for your life or run for your life or like stress out about an AP exam. One part of the response, there's a typo there, is to hydrolyze glycogen into glucose. Now, if you recall from Unit 1, glycogen is a polysaccharide. It's a stored carbohydrate, and we're going to hydrolyze that, break it down into glucose so that you have some ready-to-go glucose to make some ATP, and so that you can have a lot of energy to do your thing. Epinephrine binds to an epinephrine receptor. This is step 1, which is a GPCR. GPCR means that it's a G protein coupled receptor, right? And so here it is. Here's the epinephrine binding to the GPCR. When epinephrine binds, it triggers the phosphorylation of what's called an alpha subunit. All right, so here's the G protein. There's 3 subunits. Proteins have subunits sometimes. Go back to unit 1. This one has an alpha subunit, a beta subunit, and a gamma subunit. We don't really care about beta and gamma. We care about alpha. Alpha is going to get activated, phosphorylated by GTP, which is similar to ATP, in order to produce a response. Okay, and the alpha subunit kind of detaches from the other 2 subunits of the G protein. And what it does, alpha subunit activates an enzyme called adenylyl or adenylate cyclase, which I kind of mentioned before, converts ATP into cyclic AMP, ATP into CAMP, which is a second messenger. And CAMP molecules are produced en masse by adenylate cyclase or adenylyl cyclase, amplifying the epinephrine signal. Okay, so we're going to talk about one result of a cell receiving epinephrine. It's going to start to break down glycogen to make glucose, but epinephrine can do a whole bunch of things, and this is the fork in the road. In other words, right? So this is, check it out. There's epinephrine binding the receptor, G protein activated, starts off adenylyl cyclase, produces a bunch of CAMP. So cyclic AMP. So I put a bunch of arrows here showing that cyclic AMP has produced a lot of them, right? And this signal is broadcast throughout the cell. It's not just one thing that's going to happen here. But we're going to focus on one thing that's going to happen. One of those proteins that cyclic AMP activates what we call protein kinase A. And kinases, what do? They activate other proteins, right? So it's going to get phosphorylated by CAMP, and it's going to activate glycogen phosphorylase to hydrolyze glycogen into glucose. So all this was to break down glycogen into glucose. So you have to turn on the enzyme that's going to do that, and that's what signal transduction is about, right? Bringing in that signal and getting it to the right enzyme or the right whatever. In order to get the job done. That's signal transduction, everybody. All right, so like I said before, you can bunk up a lot of different things when it comes to a signal transduction pathway. There's a whole lot of steps involved, and therefore there's a lot of places to intervene or interfere. So we're going to talk about a few of those in 4.4. Mutations and chemicals that can alter signal transduction pathways and consequently the response to the signal. So mutations, changes in proteins, chemicals themselves, can like poisons and that kind of stuff can really mess up signal transduction pathways and responses to signals, and they can cause death. Mutations can cause misfolding or mistakes in proteins, and that's what a mutation kind of looks like. A change in a receptor, a kinase or a phosphatase can amplify or inhibit another signaling transduction pathway. I think I missed that word or that phrase, resulting in various diseases. All right, so messing up a protein can have big effects. All right, we talked about that when we were discussing enzymes as well. P53, for example, there's a very special protein called P53 that's very important for regulation of the cell cycle. I forget which checkpoint of the cell cycle, but if that protein is mutated, it can prevent cell death, it can prevent apoptosis, and it can alter the cell division, and it can lead to cancer. More on that in 4.7. Toxins, poisons, and medicines can inhibit or enhance signal transduction pathways, messing up the functions of proteins in the pathway. So 2 examples from one of my favorite YouTube people, Mr. Anderson, Bozeman Science. Okay, he has a video about changes in signal transduction. He talks about anthrax and anthrax producing a signal that denatures adenylyl cyclase or binds to adenylyl cyclase, and therefore it can't release CAMP. Cyclic AMP, and it can't, broadcast a signal. That's a really big deal. And there's another one that he talks about. It's called tetrodotoxin. And tetrodotoxin, I forget what it does. This is really bad. I forgot what it does, but pufferfish have it, and the California newt has tetrodotoxin, and if you eat it, you die. But I don't know the specifics why. So somebody maybe in the comments, check on me for that or Google it or something. Okay, all right, let's talk about 4.5. We're getting into the end here. 4.5 is about feedback, okay? It kind of seems like we're taking a left turn, really, in this topic, but we're not really because in order to maintain homeostasis, cells have to communicate with each other, and this is a very important system of how cells communicate with each other to maintain or to keep an organism alive. And that's through the maintenance of homeostasis. And that's something that living things must do. It's one of the 7 requirements of being a living thing. You have to be able to maintain stable, constant internal environment and a state of balance. Okay, so nothing can be too high, nothing can be too low. Everything has to be, as I talk to my students, it has to be Goldilocks, right? Can't be too hot, can't be too cold, can't be too high, can't be too low. Everything kind of needs to be right at this set level in order for you to maintain health, okay, and wellness, okay? Homeostasis is dynamic equilibrium, right? So if one factor goes up, your body will be able to bring it back down, okay? Or if it goes too low, your body will be able to bring it back up, but it's not going to constantly be at that set level, okay? But the important part is that your body can bring it back to that set level and maintain homeostasis. That's what living things have to be able to do. Not everything is going to be perfect all the time, but going back to there is what's important. Okay, homeostasis is necessary for organisms to respond to environmental changes, and homeostasis is maintained through what we call feedback loops, which are kind of pathways in and of themselves, and those are used to regulate signaling processes. Okay, so a super good example of a feedback loop, there's a brief picture right here. The maintenance of blood glucose. If your blood glucose is too high, that's a problem. That's called hyperglycemia. It can cause cellular damage. If your blood glucose is too low, well, you don't have enough glucose, and your cells can't make ATP in the way that they should. So we need to have a nice Goldilocks even level. So if blood sugar increases, okay, there's a hormone called insulin. You may have heard of it. That will tell your liver to store glucose from the blood as glycogen and thereby lower your blood sugar. If blood sugar goes too low, okay, there's another hormone called glucagon that will tell your liver to break down glycogen into glucose and release it into your blood so that your blood sugar returns back to that set level. Okay, this is called a negative feedback loop. Here's a couple more examples. I'm going to talk about positive feedback loops here in a second. Negative feedback loops counteract a stimulus and they cause the opposite effect as a stimulus and bring a variable back to a set point. So just like I said, blood glucose goes too high, bring it back down. Too low, bring it back up. If your body temperature gets too high or too low, it's got to go back to that normal level too. So the first example I put here, body temperature drops, it's cold in here. You shiver to heat back up, right? Muscle contraction produces heat, and it's going to try and attempt to bring your body temperature back up. If your blood glucose rises, I go drink a bunch of Mountain Dew, insulin is released to signal cells to absorb glucose from the blood. It's doing the opposite, all right? It's counteracting that change. And here's the parts of a negative feedback loop. Ready? So here's how generally it works. A stimulus is detected by a receptor, right? I get cold. Okay, wow, my body recognizes that it is cold. What's it going to do about it? Okay, the sensors that are going to, temperature receptors, like say in my skin, are going to send a message to my brain, particularly a part of my brain called the hypothalamus. And the hypothalamus is going to say, is it cold enough for me to do something about it? Do I have to do something about it? Is it below what we call the set point? Is it too far below? Is it dangerously low? Do I have to do something about it? Okay, and if it is too low, all right, and the evaluator has to do something about it, or the control center has to do something about it, it's going to send a message to an effector, and an effector is going to cause a change that is going to reverse the stimulus, right? So my muscle, it'll tell my muscles through through hormones or through long distance or synaptic signaling, it'll tell my muscles to start shivering. It'll tell my blood vessels to constrict, to conserve heat as much as I can. And so as I said, if a variable is too far from the set point, the control center signals an effector to act in response. All right, and here's our insulin and pancreas example again. All right, positive feedback loops work in a similar way. Okay, they're kind of the opposite of negative feedback loops, but they both are used to maintain homeostasis, just in a different way. They work in a similar way, but instead of counteracting a stimulus, they accelerate or amplify the initial change. They are used to achieve an outcome or do something fast. Okay, so why would you think about this? Like, all right, a positive feedback loop, why would you want to go further away from homeostasis? Why would you want to amplify a change? Well, if it's something that needs to get done, right? So one of the main examples of positive feedback is through, breastfeeding, lactation, right? A baby needs milk, a baby needs nourishment, and when a baby suckles on the areola, it's going to trigger a response in mom to produce more milk, and more milk causes more suckling, and more suckling produces more milk, and so on and so forth, and it's kind of what we call a vicious cycle. Childbirth, okay, is another example of positive feedback. Okay, I'm gonna let you look that one up. Because we're running a little short on time here, okay? But positive feedback loops are used to get something done fast, all right? And there's another example with the apples and the fruit ripening, right? Ethylene, I will talk about this one. Ethylene is going to cause fruit ripening. And guess what? When fruit ripen, they release ethylene. And when ethylene is released, it causes more fruit to ripen. And that's going to cycle over and over again. That is another positive feedback loop. Okay, so let's get to the cell cycle. The cell cycle is the life of a cell from the formation to division. So this is like part 2 of the unit. All right, the first one is all about communication. This is all about the life cycle and the reproductive cycle of a cell. And you can break down the cell cycle into 2 main phases: interphase and mitosis. All right, and hopefully you did a mitosis lab in AP biology, or you will, that analyzes a sample of tissue, how many of them are in interphase, how many of them are in mitosis, and do some statistical analysis on whether or not the amount of cells in mitosis is higher or lower than normal. Okay, but 85 to 90% of a cell's lifespan, it's in interphase, and we can categorize interphase into 3 different parts. Gap 1 is right after a cell divides. It's the first kind of part of its interphase. It grows, it performs its functions, it does its cell things. And then once it receives a signal, or once it receives the go-ahead, okay, it can go into S phase, and that is when DNA replication occurs. Okay, what is DNA replication? It is copying. Why do you need to copy your DNA? Well, mitosis, everybody, is all about a cell making a copy of itself and splitting into 2 exact identical, what we call daughter cells. And if you're trying to make 2 identical cells, well, you have to have identical DNA. Hence, this is where the copying comes from, the DNA copying, and that happens during S phase. What follows S phase is gap 2. Gap 2 is when cell prepares for division. And here's the mitotic phase here. M stands for mitosis, C stands for cytokinesis. And we're going to talk about those in just a second. Okay, but, all right, I added a little loopy on this cell cycle. Why? Because it's missing gap 0. Hey, if a cell is told not to divide, it's amitotic. It'll stay in what's called gap 0 phase. So for an example of a cell in a gap 0 phase is a neuron. You got 86 billion of them up here. They're so busy doing their job, sending and receiving signals, synaptic signals to each other, that they don't need to divide. They don't need to divide. Okay, this just doesn't make sense as part of their function. So they're in gap 0, and nerve cells will not divide. That's right, the number of brain cells you have is not going up. All right, but the important part here, well, the most important part for our purposes, is M phase, which stands for mitosis, and that mitosis is the division of the nucleus, and therefore, yes, this only happens in eukaryotes, right? Prokaryotes, called bacteria, go through something called binary fission. We might get into that later in this class, okay, but probably not too much detail. M phase is all about mitosis, division of the nucleus, and that ensures the transfer of genes to genetically identical daughter cells. Now, that is key. We're going to talk about meiosis in Unit 5, where the cells it produces are not identical, and they're not supposed to be. Mitosis, they are supposed to be identical, genetically identical. They have the same genes. And this is the type of the division that occurs in somatic cells, so regular body cells, not sex cells, not sperm cells, not egg cells. This is used for somatic cells to grow new tissues and repair old ones. That's the purpose of mitosis. Okay, I want you to know that for sure. Okay. Okay, so now we're going to talk about the phase of mitosis, right? First one that I have on this diagram is not a phase of mitosis. Why? We're in G2 of interphase. It is still interphase. And here's what's happening. I got it. I know I don't like having the pictures go off of my little page here, but whatever, I guess. So the nucleus contains the nucleolus, which is the little nucleus. That's literally what it's named. That's how you can tell that it is not in mitosis yet. The nuclear envelope, or the membrane, is enclosing the nucleus. It is fully intact, and chromosomes have not condensed. What's a chromosome? We're going to find out in a second, because they start forming in prophase. Okay, so this is what I like to kind of compare it to. Prophase is the first phase of mitosis, right? And if you're moving, let's say you're moving from one house to another, or in one apartment to another, what do you have to do with all of your stuff before you move? You have to pack. You have to pack it into boxes so it's easily transferable from one house to the next. That's what's happening during prophase to DNA. DNA is getting packed into boxes. More specifically, it's getting coiled, like very tightly wound around proteins called histones. And it is, but either way, it is getting condensed, so it is easier to ship from one cell to the next. That's what's happening during prophase. That's the main thing, okay, for prophase. That's the first part of mitosis, is that we have to get the DNA ready to split, all right, and therefore it needs to be condensed. All right, it's been copied, right? That happened during S phase. Now it has to be condensed. Okay, so during prophase, condense of DNA into chromosomes, and that's chromosomes are condensed DNA. The nucleolus disappears, and the mitotic spindle begins to form. This yellow thing over here, with the centrioles, the aster, and the mitotic spindle, the microtubules forming the mitotic spindle. They are going to start forming in prophase, which is followed by prometaphase. The mitotic spindle goes to the opposite ends of the cell. The nuclear envelope fragments, right? There's no more nucleus. Basically no more nucleus in prometaphase, so that the chromosomes can get out, so we can split them up, right? Chromosomes condense further, and the spindle attaches to the chromosomes by what are proteins called kinetochores, right? Which are going to be actually moving these things apart and splitting them up. So prometaphase is followed by metaphase. Centrosomes arrive at the opposite poles of the cell. Chromosomes are aligned along the metaphase plate or the equator, and this is key here for a checkpoint, okay? And we're going to talk about checkpoints here in a second. The cell, before it goes on to phase 5 and phase 6 here, it has to check, hey, are all the chromosomes condensed properly? Are they aligned properly? Is the spindle attached to the chromosomes? Is everything set to go before we start splitting? And that's what happens during metaphase. So here's my spindle. It's attached to all of my chromosomes through the part of the chromosomes called the centrosome or the centromere, and it's about to split them up. Ready? And that's what happens in anaphase, which is the shortest phase. Sister chromatids, aka the copied chromosomes, are separated and they move towards opposite ends of the cell. The cell elongates. This is the shortest phase because they're doing the splitting here, and that is followed by telophase, which is the last phase of mitosis, where the daughter nuclei, okay, the nuclei begin to reform. We split up that DNA between the two cells. DNA needs to be contained in the nucleus, hence the nuclei start to reform. The nucleoli reappear, the nuclear envelope begins to reform, spindle disintegrates, and mitosis is complete. All right, so check it out. We have two almost cells here. When they actually become cells, it's called cytokinesis. That happens kind of towards the end of telophase and after telophase, where the rest of the cytoplasm, so all the other stuff in the cell, gets split up between the two during cytokinesis. And that involves the formation of a cleavage furrow, okay, which is kind of like a pinch between the two plasma membranes of the cell, and pinching off the two daughter cells by the plasma membrane. So that is cytokinesis. All right, last topic here, okay. Now, that was the cell cycle, right? Interphase means it's doing its thing, it's copying its DNA, it's getting ready to divide, and the mitosis is all about division. That needs to be regulated and that needs to be controlled, because if it happens too much, that's a problem. If it happens too little, that's a problem. Hey, that's going back to like feedback a little bit, isn't it? Yeah, full circle moment, full cycle moment. So timing and rate of cell division are crucial for normal growth and development of cells. The cell cycle control system is what it's called. That regulates progression through the cell cycle, and it's a set of molecules that coordinate and triggers key events in the cell cycle. Hey, guess how they do that? It's through signal transduction, right? Wow. Again, full circle moment. Wow, amazing. That's why they put this together in the AP Biology curriculum, right? But that's what the cell cycle control system does. Now, the main players in the cell cycle control system are proteins called cyclins and cyclin-dependent kinases. There's kinases again. What do? They activate other proteins. They phosphorylate them. And they're the main proteins involved in the cell cycle control system. Signaling pathways that make a cell stop or continue through cell cycle stages are called checkpoints. So cyclins and cyclin-dependent kinases are involved in signal transduction pathways that will tell a cell, hey, you're good to go on to the next phase of the cell cycle, or no, you're not good to go on to the next phase of the cell cycle, and you should apoptose, in other words, okay? That's one of the main fail-safes of regulating cell division. Okay, there's 3 checkpoints, all right? I'm not sure if you have to know these, but I'm going to tell you anyway. The G1 checkpoint ensures the cell is large enough and the environment is suitable to divide, okay? So the cell will stop here and they will have a signal, okay, that'll say, hey, you're good to go, the environment is suitable, or no, you're not good to go. Checkpoint 2, or G2 checkpoint, ensures DNA has been correctly replicated. It checks, hey, are we good? Is the DNA copied correctly, or no, is there an error? And then finally, the M checkpoint, or the mitosis checkpoint, as I mentioned before, ensures chromosomes are attached to the spindle and kinetochores before anaphase, right? So right between metaphase to anaphase, it checks, hey, is everything aligned properly? Are we attached properly so that there's an even split? Okay, that is one more checkpoint. When checkpoints fail due to disruptions in cyclin and cyclin-dependent kinase pathways, cells can divide uncontrollably and cause cancer. Okay, so those checkpoints, right? those checkpoints and those cyclins and those CDKs, if there's something that goes wrong with them, another callback to earlier this unit, if there's something that goes wrong with them, there's a mutation in a protein like P53, for example, or there's some kind of mutagen that's messing up our proteins or interfering with the signal transduction pathways that will cause checkpoints. That is a problem, and that can cause cancer. Cancer is when cells divide uncontrollably, and they're just kind of, all gas, no brakes through the cell cycle. Just divide, 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 right? And they're sending itself signals to make it divide. So cancer cells, 3 things that you have to know about cancer cells is that cancer cells can grow without growth factors. And a growth factor, everybody, is a signal that tells the cell to divide, right? There's another example of a ligand in cell communication, right? Cancer cell will grow and it will divide without being told to. So it'll just split on its own. All right, it's just going to split. That's just what it's going to do. It's uncontrolled, right? Also, cancer cells evade apoptosis, right? So one of the fail-safe methods, I put in a picture over here. One of the fail-safe methods for preventing cells from dividing uncontrollably is just basically telling it to die. That's what apoptosis is, right? A cell will send a signal to tell another cell, hey, you are too damaged. You're dividing uncontrollably. It's going to cause problems like die. It sounds really bad, but that's what it is, right? It'll tell a cancer cell, hey, your contract is terminated, and usually that works, right? So you might have heard an interesting fact before that, like, you have cancer cells in your body that your body kills every day. Yeah, that's true, because apoptosis usually works, right? So cancer cells, once they are cancerous and they're bunked up enough, they can evade apoptosis. And the third thing about cancer, which is one of the terrible things that makes it so terrible, is that it can metastasize. So metastasis is when a cancer cell gets access to the bloodstream or it gets access to the lymphatic system, and it can travel to other parts of the body and spread in other parts of the body, right? And a tumor that is able to metastasize, it is called malignant. A tumor that cannot metastasize is called benign. So yeah, this is what cancer is all about. All right, that was it for this unit. Please let me know if you have any questions. I hope this was helpful, and we'll see you for the Unit 5 recap. Bye.